So when we eat, we get energy from three sources. One is carbohydrates, which we looked at. The other two are lipids and proteins. So what we're going to do is we're going to look at the degradation of those two other sources. Now, sugar. Where is the potential energy in sugar? Where is it located? You did all of that process, you broke it all down, and where was the energy? Because that's the potential energy. We eat sugar for energy. Okay, so where is it? That's what it's going to become, but sugar doesn't bring ATP. Where do we get the energy? Where in the bonding? We actually even have to start off and add phosphates. So sugar doesn't come in. We don't eat sugar with phosphates on it. What did we do to the sugar? What was the main reaction, the ultimate overall reaction? Not little steps, but just like that big picture. Sugar plus what yields what and what? Sugar plus oxygen yields H2O and water. Yeah, carbon dioxide and water. H2O and water, that's great. Hold on. So, we are combusting the hydrocarbon to get energy. Now, so where was the energy? Well, as we went through all of the reactions, what we were doing was we were oxidizing carbon. And so what we were doing was we were extracting the electrons that are in the carbon or around the carbon, and we did that by taking hydrides away. Okay? And we changed those hydrides to electron carriers. And that was where most of the energy came from, and it went from the pool of the carbons and the hydrogens and the oxygens that were bound into pools of, what are our two electron carriers that was used in this process? NADH and FADH2, which then went through the electron transport chain to create a gradient, which was where the energy lied, and then that gradient came back and made the ATP. All right. So when we're going to degrade fats and proteins, we're going to do similar things in terms of we're going to extract hydrides. We're going to move electrons. And we're going to take whatever we start with, and we're going to end up with, hopefully, CO2 and water as our waste products. When we get to protein, we get another waste product. What other waste product do we have to watch out for in proteins? Oh, good. We'll leave that for later then. All right, when I get to it. All right, but in terms of fats, all you have is carbons, hydrogens, and oxygens. So we're going to react that with oxygen, and we will get CO2 and water. All right, where do you get the fat to the muscle for us to actually use this? Well, first off, we eat it, okay? And so we can mobilize fats in through the diet. And what will happen is you eat the fat, and it gets into the stomach, which is an aqueous system, a very powerfully acidic environment, right? And it sits there, and it will do nothing if it doesn't get something to help it mix. And that's where the gallbladder comes in, and the bile will come in and make it soluble. And we will send it into the intestine. There is very little digestion of fat in the stomach. The acid treatment and the length of time that is there really doesn't break it down very much. Only when it gets into the intestinal line are there proteins that will break it down. The proteins that break it down are called lipases. Lipid aces, they will break down the lipids. And so it is a mechanism that occurs at a pH that is fairly neutral, but it is enzymatically cleaved. Just like everything, you can't bring in a fat molecule by itself, but we bring in the pieces, we bring in the individual fatty acids. We can transport the glycerol, And then once it's absorbed, we re-synthesize it back into a fat molecule and place it into a carrier protein. We started talking about the chylomicrons and other carrier proteins where the fats sit in the middle and I have a lipid around and proteins to target it to certain tissue types. Eventually, if we get lots and lots of fat in our diet and we don't need to do any work or very little work, like the hot pocket that comes in as we are sitting doing video games, the target molecule where all this fat goes is into adipose tissue. All right? And so, I know it gives you good eye-hand coordination, good spatial relationships with doing video games, but it is not very strenuous. All right? I don't care what those YouTubers say. It really isn't. All right? And so it sits into the adipose tissue, although it can be absorbed directly into the muscle to be used as an energy source. Now, once it has been in the adipose tissue, before it can be released, we have to give a hormonal signal to have it released. The hormonal signal will upregulate several enzyme pathways, of which we are going to now get a triacylglycerol lipase, one that is inside the adipose tissue. It's hormone sensitive, and it will break out free fats. But free fats, again, are very hydrophobic. They do not like to be transported in the blood, just floating around. So what they have to do is bind to the serum albumin, and that serum albumin has pockets that are very hydrophobic, and they can bind up to 10 molecules. When they are mobilized in this form, they are being carried directly to and interact with the muscle cell where those fatty acids are transported inside the muscle cell. Once inside the muscle cell, the fate of that fat is forever down one pathway. It will never leave the muscle cell. It will only be broken down if energy is required. Fatty acids, if you take a look at a certain what we call the typical individual, the normal individual who weighs 70 kilograms, they store energy in lots of different ways, but in terms of the value of energy, the largest single value is the fat stores that someone has. The protein, although potential there of 25,000 kcals or nutritional calories, is tied up in the muscle, and you will never mobilize all of that. Glycogen, we talked a lot about glycogen, right? We spent a whole, the whole glycolysis and the regulation and how it broke down and how it was synthesized and where it was and the liver, how we treated it, and the muscle, how we treat it. Look how much energy is there. It's 600 of that whole. And then the glucose that is in the bloodstream and in our cells that are just mobile, which we spend a whole lot of time on, is only 40 kcals. Not a lot. So there has to be a balance between this energy source, which is quickly able to be broken down, and these energy sources, which take a lot longer to break down, so that we can balance this out. So there's a lot of energy stored there, okay? And it is anhydrous, meaning that I can have a lot of energy without a lot of weight. We also have more energy in a fat than we do sugar. Yeah?
So how does this relate to like the working out for more than 30 minutes at a time?
Yeah.
So like glucose only has 40
Yeah.
calories in it, and glycogen has 600. I'm just wondering like where it flips over.
Yeah, it flips over at about, depending upon your conditioning, somewhere between the 15 and 20 minute range. Where it flips over. And so what we'll do is we'll use the ATP floating around initially. The next largest source of energy that is quick is phosphocreatine that is in your muscle. And that restores the ATP very, very quickly. Some people who do workouts use creatine. They buy a big old bucket of creatine, right? And they take 8 scoops and put it into a glass of water and they drink it down. What they're trying to do is increase the phosphocreatine so you can exercise longer. Once you start doing that, then you're starting to mobilize and start to get the sugar levels down a little bit. Well, that sugar down just a little bit will then start the hormonal signals to let the liver give off its sugar and let the muscle start using the glycogen in its stores. But that only lasts for a certain period of time. Once you get and sustain that lower level of sugar in the bloodstream because you're working out, that starts causing the hormonal signals to go to the adipose tissue. What you do is you start mobilizing the fat and you mobilize protein sources at the same time. And so by the time you're shifting from sugar sources first to the muscle and the fat, if you're well conditioned, you use about 50% muscle and fat, and then we start almost exclusively within the muscle, stop using sugar at all, and we keep that in the bloodstream. And so the muscle, once muscle has sugar, it won't ever let it go. It can use it, but once I've used that source, I want to start keeping the others coming in. And then we transition the liver, then sending out the blood glucose for the brain and the red blood cells. And so if you work out a lot, that transition happens faster. You have more of the muscle. Then you have also more of the enzymes, and you become more sensitive to the hormonal responses. And so that transition can happen faster. If you do anaerobic workout, you can increase muscle mass. You can prevent any caloric intake from coming in to be converted into fat. You just don't typically lose a lot of fat. You don't burn the fat doing it that way. You can increase the full muscle mass, and over time, just as a, you have an average calorie count that you can eat, right? Well, that's based upon how much muscle you have, and there's a basal rate at which you use energy. And so if you increase your muscle mass over time, you're not going to lose weight in the workout, but you may lose weight over time because you're just using more because you have more muscle mass. All right, fat. Oh, lots of energy. Yeah, that's where we're at. How much energy can we actually get from that? Well, that's what we're going to take a look at. Now, the lipases that we talk about, and this could be general, we saw lipases in both the intestine, and we saw lipases in the adipose tissue. They're both regulated differently, but they do have similar mechanisms. They will hydrolyze the ester groups to create the the alcohols, and then the fatty acids. All right, and so that's just adding water at the ester. The glycerol is the only portion of the fat that could be resalvaged and created into a glycotic or a glucogenic intermediate. And so just a very few parts of those. But if I want to make more fat, I'll have to get that from sugar itself. And so under extreme conditions, you can make glucose, but you're not going to make a lot of glucose even if you break down a lot of fat. When we started doing fat degradation or trying to figure this out, I really, really wouldn't want to be the graduate student, okay? Because what they did was they fed dogs different organic compounds, and then they collected what was left, okay? So you feed them, then you got to collect the urine, then you got to purify what was left over. All of that work, so one professor in his office could take credit for all the work that you just did and figure out how fats are degraded. Now, this is a short little fat, okay? Where does it not become the typical fat? Okay, typical fat has an acid, right? And has a methylene, and it has a methylene, and do we ever have a benzene ring? No. We expect that this probably would not be processed by the body, the benzene ring, okay? And that's what they intended it to be. And when they fed this particular fat to the dogs, what we had left was benzoate, okay? And you may have seen or heard of sodium benzoate. If anybody has any processed food, if you look on the label, you will probably see sodium benzoate. It's a preservative, So it's not necessarily that bad that the dogs had this. Now, we may not be able to do this exact experiment now as when they did back when. Here's another fat, and it's a little bit different. I added a carbon to it, and here I have the end product. And looking at those two different end products, if I go back just a second, I have 1, 2, 3. It seemed that this carbon that is next to the benzene ring at the end became an acid, where the acid was over here. And then over here, this carbon that is 2 away from the benzene ring became an acid, and I lost 2 carbons. Well, why in the world? I have 2 carbons here. Why am I not going to break these 2 off? Well, it's all because to get these 2 off, I have to react this particular carbon and create an acid. If I wanted to take these 2 off, I would have to react with that. That carbon does not look like this carbon, and so it isn't reactive. So we determined that this is the carbon that the enzyme works on, and it is from this acid, not alpha, but in the which position? Beta. And I go from fully reduced to almost fully oxidized, and so the degradation of fats is beta carbon becomes oxidized, or beta oxidation. Numerically, it is the 3rd carbon, where number 1 carbon is the acid, 2 is the alpha, 3 is the beta, but we refer to it as beta oxidation because that is what they named them before. All right, with fats, we have alpha, we have beta, and we have, what's that? Don't tell me it's W. It's omega. Have you ever heard of omega fats? Okay, that's where that designation comes from. All right, degradation. Before we can degrade anything, we have to activate. Okay, and so this is the activated species. Okay, with your partners at your table, correct this mechanism and add the reagent that is missing. Okay, correct the mechanism as drawn and add the reagent that is not seen on the slide that needs to be there. All right, is there anybody, any group willing to correct the book? Do you want to crack the book? Come up here, let's crack the book. Let's use red because they're wrong. Okay. Choose one place on here that you would correct.
Okay.
Yes. Before it can attack, you have to take off the proton. That is absolutely essential. Now, where does that base come from? Thank you. Where does that base come from? Randomly out of the air magically? Inside what? It's inside the enzyme as one of the amino acid side chains in the active site. Does anybody know a place on this slide that they want to correct up? Do you feel confident? Oh, come on up if you're feeling that confident. I saw it. You were drawing. Come on up. Okay, any place. There's lots to correct up around here. Ah, yes. Perfect. That's it. Yes, when you attack, you have to kick up as a transition state. Tetrahedral has to kick down, and the leaving group is AMP. Any place else that someone feels like they can correct or add a reagent? Oh, come here. You opened your mouth. When the fatty acid attacks, yes. Good. All right. Any place else that You need to add something. Oh, I saw you. You pointed. Come on up.
No, you just add water.
Where? Oh, just show me.
I don't know. I'm not sure how it reacts though.
You're not sure? I didn't ask you how it reacted, did I?
Okay, well.
That is perfect. And so that last mechanism is deprotonate the water that is supposed to be there. And I'm going to attack the phosphate. Do I attack the first one or the second one? Why? They are symmetrical and they are equivalent. And up, down, and out to break the two PIs. All right. How in the world did a publisher let this slide go by with that much wrong to it? Okay. Big picture. All right. To activate this particular molecule, I have to add CoA onto it, and it is a carbonyl with a long fat. Okay. It was an acid. To do that, I used what reagent to do the activation? Ultimately, I used ATP. But at the end, I got AMP. So I had to use a lot of that ATP, a lot of the energy to get this done. Now, have you seen something attached to CoA before? In anything that you've seen? Acetyl-CoA. What is the difference between this right here and acetyl-CoA? What the R group is. The acetyl-CoA is a methyl. An acyl-CoA is longer than a methyl. Okay? By placing a molecule on the CoA tends to take whatever it's carrying and gets it to the degradative enzyme pathway. CoA is kind of an address that carries it to the enzymes that break things down. So now I have destined this particular molecule for degradation. Okay? So I have to activate this. I add that CoA on. I'm now got ready to break it down. But the problem is it gets put on outside the mitochondria, and we do all the degradation on the inside of the mitochondria. Bugger. What do? It can't just diffuse through the membrane. How do we get through a membrane? We have to have a what? A transporter. A molecule that binds it and transports it. That seems reasonable, right? But before that happens, I just spent equivalent of 2 ATPs to put on a CoA, and the first thing that happens before it goes into the mitochondria is I take off the CoA. That seems really, really smart. Now, why I do that is because the acyl-CoA doesn't bind to any transporter. But if I take carnitine and I react it, and CoA comes right back off, I get acyl-carnitine. Now, acyl-carnitine has a funny thing that can happen. It can bind to a transporter. And so what we see at the top is the acyl-CoA, okay, and this one they actually use the 16-carbon, will come and react with an enzyme called carnitine acyl transferase. Does that seem like a reasonable name? I'm going to transfer it to a carnitine. And right behind this that says what? 1. Puts it on. I do that so that I can bind to the transporter and transport it in. Classify this kind of transporter for me. Antiport. One thing has to go in, one thing goes out. What goes out when that goes in? The carnitine molecule. That keeps that balance of carnitine Then the first thing that it sees is this enzyme, carnitine acyl transferase. What is that? 2, to put it right back onto the acetyl-CoA. They are version 1 and 2 by localization, where they are placed, and the concentration of what you have drives the reaction in one direction or the other. All right. And so now I have the acyl-CoA on the inside. I am now ready to degrade. 4 easy steps. That's just 4 easy steps. First step, oxidation. Second step, hydration. Third step, oxidation. Fourth step, what is that? Dialysis? What in the world is dialysis? What's lysis? To break. What's a thio? Sulfur. We use sulfur to break it. Okay? So let's do those 4 reactions. Okay? All right. I am going to do something scary for me. I'm going to use color. For the colorblind professor. All right. What do you have to use in a mechanism for any organic reaction inside an enzyme? You always have a what? Acid, and you always have a base. And normally, what is the first thing that you use? A base, because it's all about the... Okay. Blue or red? Talk to your neighbor. Make sure that you have a good rationale. Blue or red? Am I going to deprotonate at the blue or the red? Have an organic rationale why. Feeling it? You've got at least a 50-50 chance, right? And if you put your brains together, you have a 50-50 chance. Right? Okay, what you say? Red. Okay. All right. Let me draw the mechanism, and then we'll talk about if it's right, because I can erase. Okay. Red. Red. Blue. And you said this happens. All right. Lots of people said red. After the first arrow, what could possibly happen when you take that proton off? You could have, just for a moment, a negative charge on a carbon. Carbanion. Not necessarily what you would like, but you have the possibility of taking this and forming a resonance structure. What do we say about resonance structures? They really stabilize things, right? So having a possible resonance structure allows the red ones to be more acidic. You know that alpha to a carbonyl, the protons are acidic. If I have to deprotonate, the enzyme is going to use the fact that they are more acidic to be the ones that you're going to take off. I have now created a intermediate that is not any shorter, but a unsaturated fat. But what kind of unsaturated fat? A trans fat. We said trans fat were really, really bad. We're supposed to have cis fats. Okay, we do see a trans fat in the degradative process. But is this the stopping point? No. We have done one of how many steps? 1 of 4. Let's go on. The next enzyme is a hydratase. It's going to add a water. All right. And I think we can get this one done. If I put a base, what am I going to do with the base? What am I going to deprotonate? I'm going to deprotonate the water. if I look at my starting material, I got 1 proton, right? I look at my ending material, I got 2 protons and a hydroxide. I didn't do anything to this, but I really did something to that hydroxide. It was a water. When I take off the proton, it is a hydroxide. This now has a pair of electrons, okay? It will attack here. Why? Because that's the carbon it's on, the oxygen and the product. Then I will always have an acid somewhere available, and that picks up. But in the intermediate step, at this step, you could end up with a carbanion here, and we said that was okay because it could resonate. Okay, so that doesn't violate anything organically in terms of stability. All right, I have a hydroxyacyl-CoA. All right, I'm going to go back one step. We are doing reactions, and we are going to do oxidation at the what carbon? The beta carbon. There is the 1, 2, and 3, or the alpha and the beta. I put the oxygen on the beta. That's pretty good. All right, NAD+. Okay. You have 2 choices. A red one and a blue one. Which one is taken? I will place no particular base on the slide. Just a random placement on the. Deprotonate this. Having a negative on an oxygen is high in energy, but not that high in energy. And I will form the double bond. Not there. I will form the double bond there. And you wouldn't even have the hydrogen that's in blue. It would be the yellow one would be the other option. And what actually happens is it goes there, and that would be yellow. And I have a keto-acyl-CoA. All right, what was the first step? It was what? What did we do? Oxidation. That means we had an electron carrier. The next step we had hydration. We added water. The third step we had oxidation. We had an electron carrier. And the last is a thialysis. And the thialysis is using CoA. So I deprotonate the sulfur. That allows a sulfur to be the nucleophile. This is why I'm going to break a bond using sulfur. I will attack it here because I need a CoA on each one of the carbonyls, and one already has one. What must I do? Kick up, and then I could kick down, and then this bond picks up a proton. Acetyl-CoA leaves, and I have an acyl-CoA that is too short. All right. After this process, what have I produced? I have produced what? What is usable? Acetyl-CoA. I've also produced a FADH2, and I've also produced an NADH. Where did I produce these? In where? Yeah. Which organelle? The mitochondria. And remember when we made those types of things, where does the acetyl-CoA go to? In the mitochondria. The which cycle? Citric acid cycle. At this point in time, we took pyruvate and made it acetyl-CoA. We just took fat and made it acetyl-CoA. Does the citric acid cycle know where it came from, fat or from sugar? It doesn't care. All it wants is acetyl-CoA, and it will enter in the citric acid cycle. All right, so I make my fat, which is so many carbons. What is the most likely length of carbons? C what? C16. I did beta oxidation, and I got a C14 plus a C2. The C2 was acetyl-CoA. So how many times would you need to react the C16 to get all C2s? How many times? 7. Wait a minute. 16 divided by 2 is 7? What? Wait, but, okay, hold on. Let me think about this. C16, and I break it down once. C14 plus C2. And then I break it down again, C12 plus a C2. And I break it down again, C10, right? Then I break it down, C8. Then I break it down, C what? 6. Okay, good. We're on the same track. And then we go to C4. And then we go, you get 2 C2s. Because the 4, whatever's left, is a C2 also. So 1, 2, 3, 4, 5, 6, 7. Okay. Now, I introduced 4 enzymes, okay, to do those 4 steps. The first oxidation is really kind of specific. There is a long chain version that has higher affinities for longer chains, medium chain, and a short chain. But the last 3 enzymes are all generalized. It doesn't matter how long they are, they can still process. There are some diseases associated with long, middle, and short-chained enzymes in which you may not have one of those functional. Which is the enzyme that you absolutely don't want to have that is misfunctional? And which enzyme could you get away with having it misfunctional? Talk to your neighbor. Of the enzyme, I'm going to do A, I'm going to do B, and I'm going to do C. Which one could you live without if you had to, and which one would be really, really bad for you not to have? A, B, or C? Classify 2 of them. So, need, don't need as much. Like, I could put might die, might live. All right. Ready to give me an answer? What do you guys think? 50-50. Oh, no, 33, 33, 33. A, B, or C? What's your group? Long chain for need or don't need? Long chain you need. So they're going to put A up here. Okay, they're going to put A. Oop, they're going to put A. What about you guys? You're going to put A for need? What about you guys? A for need? What about you guys? A for need? Are you going to go with the flow, or are you going to?
I was thinking, let me just get my reasoning for it. So the medium chain spans both the short chain and the long chain.
Yes.
I would think that you would be more than likely.
Oh, we got 2 B's. 2 emphatic B's. Someone supported my option. How about back here? What did your group say? You said A? Last? You said A? All right. Here's the deal. Group B can only bind to 14 to 4. Okay? Group A can bind from 12 to 18, and Group C, nobody ever said Group C. Okay, so that one we could live without. if we had to. Here's the deal. What is the most often found carbon chain? 16. If you don't have A to degrade it at least to 14s, B working fine with no substrate doesn't keep you alive. Okay? And so we have very few that are 14 or less that are inherently there. Okay? And so this is something, unfortunately, that very early in life, so infants, they failure to thrive, and infant death is associated with discovering that they had a deficiency in that particular gene, especially when they are on a diet of milk as their primary source. So long chain is much, much more important to us than the short chain. All right? Got it? Okay, good. All right. Last slide I'll cover. So the number of reactions that you need to use to degrade it is n minus 2 divided by 2. 16 minus 2 is 14 divided by 2 is 7. You will produce n minus 2 divided by 2 FADs. You will produce n minus 2 divided by 2 NADHs. I can get 1.5 ATPs for every FADH2, and I get 2.5 for NADHs. I produce n minus 2 divided by 2 protons, and I will get n divided by 2 acetyl-CoAs, which can enter in the citric acid cycle, and once fully oxidized, can produce up to 10 ATPs. Now, you do have to subtract 2 ATPs for the degradation, or for the activation itself. Okay? So, a normal 16 fat, how many ATPs can you synthesize by fully oxidizing one fat molecule, fatty acid, that is C16, that gets into the muscle cell? Bring back that number when you come back next time. All right? It may be a few. The question is, oxidize a 16 carbon fat. How many ATPs do you produce?